Solving problems involving monohybrid crosses
using Punnett squares.
Gregor Mendel was an Austrian monk who, through the experimentation in his garden, discovered the hereditary traits which were passed on to the offspring. He is now considered a pioneer in the field of genetics. Mendel showed through his experiments on peas the inheritance of certain traits, also known as Mendelian characteristics. Through the use of cross-fertilization of the pea plant with clearly opposite characteristics, for example purple and white flowers or tall and short plants, he was able to distinguish the dominant and recessive genes. The conclusion of these experiments established that the traits from the parents to the offspring are passed on independently. This is known as the law of independent assortment.
The Punnett square was created in 1905 by Reginald Punnett and is used to illustrate Mendel's discoveries of monohybrid crosses. To begin a Punnett square calculation, the parental alleles are analysed into their genotype abbreviated forms as segregated haploid gametes and are written on top and the left-hand side of the Punnett square grid. The haploid gametes are then crossed to determine which alleles will be combined. Each box in the Punnett square grid symbolises the diploid genotype of the zygote. By analysing the results of the Punnett square, as well as the dominant and recessive genes, the genotype and the phenotype can be determined.
Here's some key definitions. Heterozygous is when an organism has two alleles from a particular gene which are different, while a homozygous organism has two alleles which are identical. Through the use of Punnett squares, a genotypic and phenotypic ratio can be calculated. A phenotype is the given gene that is present in the offspring, but is not expressed in the appearance. The range of forms in which a gene can take is classified as alleles. A phenotype is defined as the characteristics which can be viewed in an organism's appearance. When documenting crosses, genes are symbolized by letters in abbreviated form. The dominant allele is represented by a capital letter and the recessive as a lowercase letter.
A monohybrid cross is an analysis of inheriting a single characteristic or trait, the crossing of true breeding organisms. In the case of a monohybrid cross, only genotype is possible, meaning all offspring will have a heterozygous dominant genotype, as one allele from each parent is given to the offspring. For example, Mendel studied various traits of the pea plant, one of which being the colour expressed on the pea plant flowers. To begin, Mendel needed plants that were true breeding, meaning after generations of self-breeding, the plant will only express a single version of one trait. Then, he crossed true breeding white flower plant with a true breeding purple flower plant. This experiment is defined as a monohybrid cross.
The F1 generation is the crossing of two true breeding organisms. This generation was crossed with the plants with identical traits. Mendel discovered that the F1 were all purple flowers. As this generation inherits the purple allele, dominant trait, and the white allele, the recessive trait, meaning the purple trait masked the white characteristic. The F2 generation consisted of a 3-to-1 ratio of purple to white flowers. These results can be explained through the law of segregation.
Brahford cattle are an example of a monohybrid cross, as they are a result of a breeding Hereford and Brahman cattle together. Brahman and Hereford cattle were bred in Queensland, Australia, to produce cattle of a quiet nature with tender, well-flavoured meat. They are also resistant to parasites and the heat, and grow at a faster rate to a large size. To illustrate this monohybrid cross of the parent generation, a Punnett square will be used. Let the capital R represent the parasite-resistant allele, which is the dominant, and the small r represent the non-resistant allele, which is recessive. When the alleles are crossed, the genotype is 100% big R little r, meaning the phenotype of the cattle will be parasite-resistant, whilst they still carry the recessive gene. This generation of offspring is called F1. If the F1 generation are bred together, the genotype will be a 25% big R, 50% big R, and 25% little r, The phenotype of the offspring will be 75% parasite resistant and 25% non-resistant.
Now we have a quick test to test your knowledge. Pause to write down the answers. What is the definition of a monohybrid cross? What is the definition of heterozygous and homozygous? What is Gregor Mendel's contribution to the development of genetics? How does a dominant allele affect the recessive allele? And what is the difference between phenotype and genotype?